Thank you very much. I think the theme throughout my talk will be applied genomics, as is one of my titles, as you heard. And I think what I will do is taking what you've heard from all the previous speakers and what we're trying to do, and much of my work I do together with my co-leader of the breast program, Dr. Laura Esserman, who actually a graduate from Stanford, in two different programs. So I'll touch upon actually two of them. One is in the area of healthy women, who is actually at risk for cancer, of breast cancer specifically, and how do we use all our knowledge and actually generate maybe new knowledge out of what all we're gathering in this 150,000 women cohort. And my second part will be on therapy choice and response. So if somebody is at high risk to develop a recurrence, how do we best treat those women? And so those are two different programs that are coordinated out of UCSF with many, many collaborators. One is the Athena Network and the other one is the I-SPY 2 trial. So for the Athena Network, I'm also working with David Hausler and David Patterson under a Big Data to Knowledge Center for Translational Genomics, where we've taken a part of this Athena study, our wisdom study, to develop a use case how to integrate big data. And the context of this use case is actually in the area of breast cancer screening. If you follow the papers, there are many different ways how women are advised, how frequently to be screened, what age to start. And women are sort of caught in the middle, and we developed with investigators from the UC system and others a clinical trial to address that. We were able to do that because our UC-wide breast health program actually includes women that are either being screened or diagnosed with breast cancer, and we integrate all of their knowledge by questionnaire systems, as well as individualized assessment of biology, germline status into clinical care. So in one slide, this large clinical trial called WISDOM, Women Informed to Screen Depending on Measures of Risk, will compare, which is mostly the standard, women being screened yearly by mammography as compared to what we call the personalized arm, where we introduce all of our knowledge into who is actually at risk at what level. And so this personalized arm, one of the arms, as said, includes the breast density of a mammogram, the health questionnaire of a person's risk factors, as well as genetic testing. And this is we're going to do in a study of 100,000 women out of our 150,000 cohort, and we will compare the risk level for each woman over the next 5 years, which then will allow us to set threshold that somebody doesn't have to be screened until age 50. Somebody can, as of age 50, have every other year screening, annual or maybe most frequent screening. And so how do we do this? We collaborate with a company called Color Genomics here in Birmingham, and we're testing currently 9 susceptibility genes in all of these women, as well as a polygenic risk score. So this is a risk score that you build out of, currently we're using 81 gene variants that are all associated individually with a little increased risk, but you can understand if you have all of these in the risk variant, then your risk starts to increase, which you can see on this very small graph on the bottom. So what do we need for big data to actually do all this? Because we're generating a lot of data on all of these women, and they're not really standardized systems to collect all this data. So what we're currently doing as a use case to make sure that we store all this data in a very secure way, and also in a way that we can have authorized access on the iDash-Cloud at the University of San Diego Supercomputer Center. And here you can see all of the data sources that we're actually gathering for all of these women, where in the middle is this iDash-Cloud, and around it, for instance, on the bottom left part, is the structured electronic medical record data. So you can understand if you have in the top right the genomics data, all of the medical record data, you can, with all of your knowledge of the genomics we're testing, we're actually doing a series of other testing in the background, we can derive new knowledge based on a cohort selection tool, as you can see on the right. So we're not yet I'm not showing you any data yet, but we've done the first pilot and we'll start actually in the summer enrolling the 100,000 women in this particular trial. I think this will generate a lot of knowledge to work on. So the next actually use case, so to speak, for big data is more data integration across many different layers. And this is in the area if somebody is diagnosed with aggressive breast cancer and you want to treat such a person with the best possible treatment to reduce the risk of recurrence, as it's mostly the distant recurrence that women will die of their disease. So one of the ways to identify women at high risk is the 70 gene prognosis signature. I was part of developing that some 12 years ago. And if somebody is high risk, you can see in the bottom part a high proportion of women develop a recurrence. And we actually recently confirmed this particular finding in a 7,000 patient trial where people were randomized to either use this or not for their further treatment, and showed that indeed if one is genomically low risk, that they can forgo chemotherapy, but if you are high risk, that is beneficial for treatment. So what are we doing in this I-SPY? We're selecting women at high risk of recurrence, and you've seen the ways how you can identify networks that are derailed, to which particular treatments, targeted treatments, are developed. And in this I-SPY trial, where Laura Esserman and Don Barry from MD Anderson are the PIs, I'm leading the biomarker work. We're investigating what are the determinants of response with these particular drugs. So this is an adaptive randomized trial, which means that there are 5 investigational arms. Shown here are 2 next to the standard arm. And once you see for a predetermined set of 8 biological signatures, that one signature shows the most response for a particular drug. So let's say you have a biological type A and a treatment A, then you would see that if the biological type A is randomized into treatment A arm, you'll see a response. And once that hits a particular threshold, you'll start to enrich for those type of patients in that arm. Thereby you can reduce the number of patients to come to a success of graduating that drug with the biomarker combination. So here is a series of, in the second column you can see what is the biology that's being targeted. We're actually many of those graduated, and we're in the next series because it's an open protocol continuously adding new drugs. So this is how the adaptive randomization works from left to right in the beginning. At first there is a random randomization, the learning phase, and patients are being typed for biological type, here indicated by purple, blue, and orange. And on the right side, if at surgery, so the treatment is given before surgery, so you can monitor response, it's evaluated by an X if the tumor is completely gone, associating, for instance, for drug number 1 that the blue type responds. So if that hits a particular threshold, then the next patient in the trial is blue get preferentially randomized to drug 1, as you can see in the adaptive phase. Again, as it assessed whether those are what is the response rate, and if that hits a particular percentage of a likelihood success in a subsequent trial, then the drug graduates. And so by enriching, you can reduce the number of patients and you focus on those biological subsets that respond. So here's the most up-to-date list of, in purple, all the drugs that graduated from the trial in a particular subset, as you can see, or here is the subtypes that are eligible, and I'll show you one example to show you what is actually then the subset in which it graduates. Since it's an adaptive trial, the probabilities for success, which is called PCR, pathological complete remission, is shown as a distribution. And what you're looking for in evaluating the drug is the distance between those two curves. And if that is large enough, larger than control standard treatment alone, then the drug graduates. So we're looking at the estimated PCR rates. I'll show you one example with a few slides for a treatment that actually attacks the DNA repair deficiency, and it's known that cancer cells are lacking effective DNA repair, some of them, and so if you further inhibit DNA repair, then the cell can no longer survive. So the question is, which tumor types actually have such a DNA repair deficiency so that this is effective, and can we identify that specifically? Just to make a long story short, if you look at the three panels here, you can see that in the bottom one, the two curves are furthest apart. So that means that if you don't take all patients, as in the left, but in the bottom, the so-called triple negative, that's where the drug is effective. So this DNA repair inhibitor graduated for those particular patients. But we're actually not satisfied with that, because in the background we do all of this data integration with different types of genomics testing, phosphoproteins, gene expression, gene mutation, and which is called the qualifying biomarkers, to see if we can further specify who are those women within that group even that respond. And for those of you working in this area, you may recognize germline status of BRCA1 and 2, a PARP inhibitor signature that we developed from cell line work, and others which we tested. And we're interested in the ones that all the way to the right continue to have a red yes here. So what we actually did, this is long work and hope soon to publish this, is that if you take those triple negative patients and you combine two of those signatures, the PARP sensitivity marker and a very high risk in proliferation, then you can see that those curves are very far apart. So now they're not from 27 to 56, but they're 23 to 79. And so that would mean that those 40% of the triple negative, that's actually where all the response is being found. So this is exploring where actually we can further, we would just to validate. So to summarize, we can, by this qualifying scheme of all of those biomarkers, find actually the highest likelihood patients, which we need to validate in the next trial, but that would mean you can really zoom into the patients who will benefit from that particular treatment, meaning that others who have low likelihood can go on to another treatment. So we can this way zoom in and enrich for those patients, which means that we accelerate to identify which drug works for which patients, and that reduce the cost, time, and the number of patients to get this to a process that FDA has now agreed upon, accelerated approval. Also this, of course, there's a lot of data, clinical trial data, patient-derived information, genomics data, and here we're also using different interfaces to actually, and we're building this part to combine all of these data to do even further research beyond our qualifying, but now go to the exploratory level. So far, 1,500 patients are screened for this trial, so this has become a very rich and huge data set. So thank you very much, and here are my genes. Thank you.